for the day. So starting off with some announcements, things going on around the core. One is we put together a document called How to Read a Scientific Paper. And so if you don't have a lot of experience, it should be really helpful for you because it'll help direct you towards like what kinds of things should you expect to find in the discussion section, in the introduction, and how to get the most out of reading a paper. It's easier than just sort of slogging through start to finish without stopping. So check that out. It should be helpful for you. General reminder, keep an eye out for a research paper to do your poster presentation on, something relevant to human behavior and development. Professor Lyons mentioned yesterday that we also have a supplemental epigenetics reading, and so check that out if you want a sort of more introductory, sort of accessible explanation of epigenetics and how we see that working in the world. It might be a little easier to understand than the other one that was assigned. And then for p-set rationales, one is that it's important to know that they do matter at the end of the quarter if you sort of end up between like B plus and an A minus like cutoff. If you did like really great p-set rationales the whole quarter, it'll help bump you up into the next grade category. But if not, you'll sort of stay where you are. And so work on those. They're helpful. I look over them every week, sort of everybody's. I just sort of like see how they went. And once or twice throughout the quarter, you'll get sort of specific feedback, rationale by rationale, student by student, on what you wrote. So look out for that. It wasn't this section this week, but it will be at some point in the future. And then if you want to come to Food Inc. this evening at 6 p.m. in HumBio in the Essay Lounge. Anybody planning on coming? It's going to be cool. That's all right. Yeah, so we'll be doing that. Yeah, question? Yeah. If you look on Piazza, I pasted like what coursework does automatically in those grading things. It's not exactly the way we wish it was, that you could get partial credit in the same way, but it is possible to get partial credit. It's just a penalty for guessing. So if you guess a wrong one, it actually like subtracts points that you earned by checking the right ones. So, because I got like,
yeah, just, like for one of them, it gave me like 0 points.
Mm-hmm. Yeah, exactly. And a lot of people were in that situation, and that's because it like subtracted the correct points. Whereas if you had like, only checked like half of the ones that you were supposed to check, but none of the other ones, then you would have gotten like half points. So it's like partial credit is kind of possible. It just subtracts for incorrect guesses.
So if you check an extraneous one, you get no points?
It'll subtract like a proportional amount of points. So if there's 5 answers and you're supposed to check 4 of them, but you check that extra one, it'll like subtract 1 of your answers, and you'll still get like 3/4 of the points. Don't check all of them. Yeah. Exactly. Yeah. And we put in a request for a change, but that's sort of like a major coursework change, so I don't know if that'll happen or not. But yeah, so that's how that grading works. All right, so for our content today, so the big focus of this course, of course, is gene and environment interaction, right? We're looking at behaviors, phenotypes, things in the world, our outcomes, our dependent variables, and we're trying to explain how did that come to be? How is it? Why is it? Last quarter we talked a lot about the dual inheritance model, where we looked a lot at the cultural aspect of the environment. And so you should be used to applying that. And today what we're going to do is unpack some of the models, Bronfenbrenner and Hertzman, and then some of the sort of different types of interactions in terms of genes and environment in the way that they influence behavior. All right. So to start with our models. So from last week in lecture, you'll remember we talked about breastfeeding, right? And so if you have your little infant in the middle of Bronfenbrenner's model, right? So this is Bronfenbrenner's model of this concentric, nested context of development. There's a lot of factors that influence whether this infant gets fed or not, right? Breastfed. So things in the microsystem and things working out to greater and greater distances all impact whether the infant gets fed or not. So what's something sort of a far-out exosystem, mesosystem sort of impact that might determine whether an infant is breastfed or not? Clean water, yeah. So it's like sort of sanitation policies for a given state or town or something like that. Do they have access to clean water or not. And that would especially impact the impact of like formula feeding, right? Because if they have polluted water and they're being fed formula, then that sort of contributes to childhood mortality, right? And you could also think about like parental leave policies. You know, is this mother in a job or in a society where she's allowed to take time off after having a child to do breastfeeding? Because it takes time, right? You can't do it if you have to go to work all day, every day. So that's sort of Bronfenbrenner's, and his model, the emphasis is on sort of a snapshot, right? So at any one given time, this infant, this developing person, what are all the factors that influence it? Now, Hertzman comes along. What does Hertzman add to this Bronfenbrenner model? Yeah, it's a life course trajectory, right? So that's symbolized by this arrow that sort of traverses the whole model. And that's sort of saying that all of these things matter, yes, but they matter sort of year by year as this person is developing over time. And so Hertzman focuses a lot on those developmental effects. And so one of the things that came up with Hertzman is this idea of pathway effects. And so pathway effects is when, you know, sort of 1 year builds on the next year, builds on the next year, and over time, something like, you know, maybe not being breastfed gives you slightly, you know, lower neural development, right? And that might mean you don't do quite as well in school. And that might mean you have slightly less self-efficacy or value for education, which might mean that you don't go to college, which might mean that you don't get, you know, the same sort of job that you would otherwise have, right? So it's all about these effects that build up over the course of a lifetime that we consider to be pathway effects. Yeah, so in the Hertzman reading, he sort of distinguished between the cumulative effects, the pathway effects, and the latent effects. And we asked about that on the PSET just to get you to do a close reading of the article, but I wouldn't stress too much about distinguishing between like what's really a latent effect and what's really a pathway effect. But he does distinguish that like a pathway effect is not the same as a cumulative effect, which is just sort of a buildup over time. A pathway effect is more like you're set on a particular trajectory. All right, so now to the first page of your handout. You see this model right here. So this is another Hertzman idea, the idea of biological embedding. So who can tell us in simple terms, what is biological embedding? Anybody? Biological embedding? Yeah, Lila.
It's like when your experiences become like physically like part of your body, like I guess embedded in some ways. That can be like epigenetic changes, or like in sculpting neural circuits at like earlier points in your life, like carried over later.
Yeah, exactly. It's about that interface between the environment and your own body. Like how does, you'll hear this phrase, like how does the environment get under the skin, right? So how does working in a stressful workplace actually, you know, change your neural circuits and your neurotransmitters, and how does it change the way that your, you know, body reacts to pathogens? You know, how does that lead to more sickness? How does it affect your actual health and development? Right. So that's just the idea of biological embedding. And so what we're going to do is take a quick walk through this model. Right. So we'll think about breastfeeding as an example, right? So that would be sort of the top thing, quality of the early environment, right? So in your early environment, were you breastfed or not? And so that leads to the second step, the sculpting and neurochemistry of the central nervous system. And so if we say that, you know, the central nervous system can be sculpted, that's telling us that there's a certain degree of plasticity, right? So that means that it can change. If a neural circuit is plastic, it can change. It could be sort of adaptively plastic in a good environment over time, or it could change maladaptively and have maladaptive plasticity in a poor environment over time. And so you'll remember we talked about different ways of studying the brain last week. What if we were looking with MRI, not fMRI, which is functional, but MRI, what kinds of neural sculpting would we be able to see? Yeah, Darcy.
It would be like the physical layout of the brain.
Yeah, exactly. And so, like, what about the physical layout? What could we compare between, one person and another person?
See how large like certain regions of the brain are?
Yeah, like size of brain regions you could see with MRI. You can also see, white matter versus gray matter, right? So you can see whether things are myelinating correctly with their white matter or not. And so we're talking about the sculpting of the central nervous system. So what about breast milk is really helpful for the sculpting of the central nervous system? What's in it? Colostrum's good. That one's especially good for immunological function. What about for neural function? Yeah. Yeah, omega-3s, right? And those were that was a specific example of, what were the big things called? Was their acronym? Yeah, LC-PUFAs, long-chain polyunsaturated fatty acids, of which omega-3 is one. Right? And that's really helpful for myelinating your neural circuits. And you'll hear tomorrow in lecture, actually, about how that's important for development of the visual system especially. You need these fatty acids to develop that myelination. Long-chain polyunsaturated fatty acids. And if this were A-side, you might look at like the structure of the chemicals and see like what makes it polyunsaturated. But for our purposes, just knowing that that's what they are is sufficient. All right, so that's great. So now that's how we might sculpt the neurochemistry of the central nervous system. And then that, in turn, is going to go and affect all this next level, right? So your endocrine system, your immune system, all of those things then, go down to impact your psychosocial and behavioral development. So that's sort of the outcome. Okay, what kind of behaviors, what kind of phenotypes do you develop at the end of this? And then there's one more sort of all-important aspect of this model, which is the looping around, right? The arrow that goes from these outcomes back to sculpt the neurochemistry of the central nervous system again, right? So it continues around and around. It doesn't just end with one output. And then one final big take-home point from both of these models and this idea of neuroplasticity is that it's easiest to intervene and change a neural circuit when it's developing most rapidly. Right, so that's typically, earlier in life the better. That's when things are really developing quickest, and if you want to direct them, impact them in a positive or negative direction, it's easiest to do that when they're proceeding most rapidly. Okay. Yeah, and we'll talk a lot more about critical periods, sensitive periods over the course of the next couple weeks with language development and things like that. But yeah, it's more of a sensitive period if intervention at that time produces a big change in sort of big outcome difference. All right, so that'll bring us to our next page here. We're going to start talking about main effects, genes, environment, and the way they interact or not. So one of the things that we're going to be looking at with all of these charts is plasticity. So that's sort of a key term, and phenotypic plasticity is the phenotype's susceptibility to change depending on different environments. So phenotypic plasticity is a phenotype's susceptibility to change depending on different environmental experiences. So what is it going to mean if we have high phenotypic plasticity?
Environment will have a greater effect on shaping phenotype.
Yeah, so the phenotype is going to be really different in different environments, right? And if there's low phenotypic plasticity, then the phenotype is mostly set. And it doesn't really matter what environment you end up in, it's set. So that would be low phenotypic plasticity. So now we're going to run through a few graphs to sort of unpack what that actually looks like. And so these are sort of conceptual graphs, just to sort of tease apart the idea of like what is a gene-only main effect and an environment-only main effect. And we're going to have 3 different variables, right? So what in these graphs is our dependent variable, our outcome variable? Obesity, right? That usually goes on the y-axis. It's sort of what you're measuring as an outcome. And then we have 2 different independent variables, right? And so you're going to see how we deal with two different independent variables. One of them is our low-fat environment versus high-fat environment. So sort of what does the environmental context look like? What's our other independent variable? Genes, right? Genotype. So we're going to have like genotype A in red and genotype B in green for these graphs. All right. And so that brings us to this idea of main effects, right? So that's the first blank at the top is like, what do we mean by a main effect? So a main effect is the effect of an independent variable on a dependent variable, controlling for any other independent variables. So a main effect is the effect of an independent variable on a dependent variable, controlling for any other independent variables. So for something like obesity, that's our dependent variable, many different things might have their own main effect on obesity. All the different independent variables that you have might have a main effect on obesity. And you'll see the main effect of an independent variable come out once you've controlled for any of the other independent variables that have an effect. All right, so our first case, we have a gene-only main effect, right? So if we have a genotype-only main effect, that tells us two things. One, that genes matter. So you're going to have different levels of obesity depending on what genotype you have. And two, if it's gene-only main effect, then environment isn't going to impact them, right? So who could come and draw sort of what our two genotypes are going to look like in a gene-only main effect? Yeah, Garcia. And we'll do them as like dots and lines as opposed to bars. Nice. Exactly. Thank you very much. And so in this case, genes matter, right? If you've got genotype A up here, higher risk of obesity, genotype B, lower risk of obesity, and no effect of environment. So in this case, is plasticity high or low? Low, right? Because we said plasticity was how different your phenotypic outcomes are in different environments. And in this case, in different environments, there's no difference in the phenotypic outcomes. And you might remember the term heritability, right? And heritability is how much of the phenotypic variation can be attributed to genetic variation.
There's high heritability in this.
Exactly. Yeah, so in this case, we have high heritability because how much of the phenotypic variation, high obesity, low obesity, can be attributed to genetic variation. So in this case, it's a lot, right? Because if you know that someone is genotype A, then in both of these environments, they're going to end up with higher risk of obesity. If they're genotype B, they have lower risk of obesity. And so genes are sort of all predictive, so that means it's highly heritable. Yeah?
Does this translate directly into why he's explaining that heritability is highest in homogeneous environments?
Yeah, yes, it is. Because, in reality, so you said in homogeneous environments, heritability is highest. So if you control for sort of all the possible environmental factors, then you're really only going to see the effect of genes. And so you usually only define heritability given a particular environment or environments that you're talking about. So we'd say in these two environments, at least, that we studied these genotypes, there's very high heritability because there's no variation with the environment. All right, so how about an environment-only main effect? So this would mean, again, two things. One, that environment makes a difference, right? Environment has a main effect on obesity. And if it's environment only, then genes don't have an effect on obesity. So who wants to draw this one? Yeah. Yeah, exactly. All right. Nice. Exactly. And so those lines are, if it's literally environment only, then we're not going to see any difference between the genotypes, right? And as you would expect, in the high-fat environment, we have higher rates of obesity. And so in this case, plasticity, high or low? High, right? So everybody think about that. Why would plasticity be high? Yeah, different environments give you very different outcomes. And what about heritability? High or low? Low, right? Because it's sort of the opposite of this graph. Here, if you knew the genotype, you had a great predictive power over what phenotype would result. Here, if you have a genotype, you don't know what their phenotype is going to look like. It doesn't tell you anything. Yeah, Lily?
Would it be true to say that plasticity and heritability tend to be always inversely correlated?
Let's see. I hesitate to say like always inversely correlated. It's a good question.
Most of the time?
Like in this case, in our cases, yes, they will be, because like the more that it changes with the environment,
Right.
that means the less it changes with genes, right? So yeah, so I think so. I might have to think about that and get back to you though about like whether it's always the case. But yeah. All right. So now we've got two coming up. In this case, we're going to have gene and environment main effects. So genes have a main effect and environment has a main effect, but they're not interacting. So that means that the way that the genes are expressed doesn't matter what environment they're in, they're expressed in the same way. And then down here, we're going to have a gene-environment interaction, which means the way that the genes are expressed really depends on which environment they're in. And we'll unpack that in about like 3 or 4 different ways. So hopefully everybody can get it by the time we're finished there. Okay, so first of all, gene and environment main effects without any interaction. So if we have a both gene and environment effect, then it matters what genotype you have. That's going to give you differential rates, and it matters which phenotype which environment you're in. That's going to give you different phenotypic outcomes. And so if we say that there's no interaction, the size of the effect of the environment is going to be the same in no matter what environment it's in. The size of the effect of that is going to be the same, regardless of what genotype you have. And vice versa. So the size of the effect of genotype is going to be the same size of effect in one environment as it is in the other environment. And I'll show you more what that means once we get our graph up there. So in this case, so let's see, A was our higher risk genotype before. So we've got our A, right? Because we know that environment has a main effect, so it matters whether you're low environment or high environment. And what about genotype B? What's this one going to look like? Exactly. So it's still parallel, but it's lower. Now again, you'll notice a few things. Like in all these cases, we sort of have parallel lines, right? So they're going in the same direction at the same sort of slope. And there's no interaction, because even though the genotypes give you sort of a different starting point, right, either down here or up here, the effect size of genotype, which is like how much difference it makes, how much difference genotype makes, is the same in this environment as in this environment. So the distance between those lines stays the same. And that's telling us that it doesn't matter what environment you're in, genes still have the same amount of effect, right? And it also doesn't matter which gene you have, environment also has the same amount of effect. So they're not interacting. All right, so now in this case, what about plasticity? Do we see a lot of plasticity going on here? Yeah, who thinks, yeah, so yes. So yeah, because in different environments we still get very different phenotypic outcomes, plasticity is still high. And what about heritability compared to these first two graphs? Think about that for a minute. Yeah, so who wants to explain, sort of unpack that? What about heritability for this graph? Yeah, Corey.
So I guess, like, depending on, like, what type of gene you have, like, you sort of have, like, an upper and lower, like, you have a range that you can get
Yeah.
depending on your genes. Like, depending on what environment you are in, like, you can reach, like, one end of the spectrum. But no matter, like, what environment you're put in, like, there is still, like, a limit. Like, if, let's say it's height, like, there's a limit to, like, how tall you're going to be regardless of, like, your environment.
Yeah, that's great. So if you know the genotype of a person, that'll tell you something about their phenotype, right? It won't tell you everything. So heritability isn't as high as in this first case, but it'll tell you something. So on average, people with green genotype are going to have lower risk of obesity than people with red genotype. But we don't know exactly how much because the environment also contributes to the variation. So it's not just genotype. So it's sort of a moderate degree of heritability. Right. Yeah.
For B, then you can is it heritable, like the second one you did, in the sense that you would still have a range of where the phenotype would fall? You just wouldn't know which genotype it was.
So in this case, we would see. You're talking about this case, right?
Yeah, because you're judging the heritability based on, I guess, the y-axis that your lines cover.
Yeah, and not just the y-axis, but how, like, where somebody falls on that y-axis. Is it because of their genes, in which case it's heritability, or is it because of something else, in which case it's low heritability? So think about this case. So here, if you know their genes, like, how well can you predict where they're going to fall on obesity spectrum? So if you know that they're genotype A or genotype B, that doesn't actually tell you anything, right? Because, you know, given all the people who have genotype A and all the people who have genotype B, they fall in the same range, and it's all because of the environment. Yeah, so think about that. And if you want to, we can talk more after class, because it's a tricky concept. And if you want, I put a little blurb on coursework under week 2 section materials that'll help you think through heritability and practice that a little bit. So again, we don't see an interaction here because the size of the effect of genotype is the same in both environments. Yeah?
We still see the same amount of plasticity as the graph before.
Yeah, so let's see. So this was highly plastic, very high plasticity. And would it be the exact same amount of plasticity?
Because, like, would plasticity be affected by the genes?
Yeah, I don't think so. That's a good question. I'll check into that and sort of see how we compare the plasticity between those, and I'll email back to the group.
Like, would it be like a degree of like the slope? Like, if B and C have the same slopes and like the plasticity is the same, but like
Right.
if it, like the slope was different with C, but like still parallel with both, would like that mean there would be less plasticity?
Yeah, you know,
Because,
if...
like, different, like the degree of variation is different.
Yeah, like if it changed more according to environment here, then you would say like probably like more plasticity. Like if, you know, these lines were really slopey, so like environment had a huge effect, like you could change a lot more, you'd say more plasticity than if there's less change between environments. So yeah, I think you're right. It is sort of about the slope of these lines.
Okay, but I mean, bottom line, there is still plasticity.
Yeah. Yeah, exactly. All right, so now eventually we're going to get to a gene-environment interaction, right? So when we say that genes and environment are interacting, what we're saying is that the environment influences whether or not a gene is expressed or not. And so the definition of the interaction is you have an interaction between two independent variables when the size of the effect of one of the independent variables on the dependent variable depends on the value of the other independent variable. And so I know that's a long sentence, so I just typed it up on the board. So we have an interaction between two independent variables when the size of the effect of one of the independent variables on the dependent variable depends on the value of the other independent variable.
You said like right before you said that sentence, you said environment is what?
So the environment is determining to what extent a gene is expressed. So that's sort of taking that sentence and applying it to our case of genes and environment. But just in general, you have an interaction between two independent variables when the size of the effect of one of the independent variables on the dependent variable depends on the value of the other independent variable. So for the example that we're using up here, you'd probably have an interaction when the effect of the environment is bigger for one genotype than, or the effect of genotype is bigger in one environment than in the other environment. So a good gene by environment interaction example is the thrifty genotype hypothesis proposed by James Neal. So this is different from thrifty phenotype, when we'll talk about in a bit. But does anybody remember what is this thrifty genotype hypothesis about?
So like there's the hypothesis authors, they didn't encounter like a lot of fat a lot.
Mhm.
So when they did encounter it, they were able to store it really well. And basically, people who were able to do that were able to like survive more, so that gene got passed on.
Totally.
So like now, because we're able to like have a lot of fat now, like people have this thrifty gene where they like store fat when they encounter it, but like it's not like we rarely encounter it.
Yeah, exactly, right? So the thrifty genotype is the genotype that, you know, for every calorie your body can get its hands on, it tries to store it, you know, hold on to it as glycogen and fat. And then, of course, in our modern environment, that's not so adaptive anymore because we have ready access to sugary and fatty foods. So genes that were previously adaptive are now harmful in our current environment, right? So this brings up the example of the Pima Native Americans. So there's their genotype. So I'll put it in red, and we're going to graph it as the gene-environment interaction. The Pima genotype and the non-Pima genotype is what we have. Okay. Now take a minute and on your own, imagine or draw what do you think these graphs are going to look like for the Pima genotype and the non-Pima genotype in the low-fat environment and the high-fat environment. And your clue is that the Pima genotype is supposed to represent this sort of older, evolved, evolutionary, thrifty genotype. All right, so it should look something like this. So you've got your Pima genotype, which in a high-fat environment has a much greater risk of obesity than the non-Pima genotype. However, again, we have our interaction between genes and environment, because in this environment, those genes don't do the same thing. This environment doesn't cause any difference between those genes. So they're essentially the same in the low-fat environment. You both have a low risk of obesity and diabetes. So in this case, we know it's an interaction because we can't say, you know, is one genotype better than the other? You have to say, well, it depends. It depends on which environment you're talking about. And just in general, when you're looking at graphs like this, if we see our lines that are parallel, then we're not looking at an interaction, right? Because the effect size is exactly the same in both cases, both independent variables. But when they're angled sort of towards each other or even crisscross, then we're looking at an interaction. So that makes that's sort of graphically what you'll look for if you're trying to see an interaction. Yeah, question?
Would we say that genes like don't have a main effect here? Or like do either have a main effect? Or is
Yeah, so you would have to, if you're going to say that like one of them has a main effect, you would have to control for the other one. So you would have to say like, okay, we're going to eliminate environmental variation. And if we look at just in a high-fat environment, then yes, there's a main effect of genes. But if we look in a low-fat environment, then no, there is no main effect of genes, right? But there is a main effect of environment because, right, in both genotypes, you get an increased susceptibility depending on what environment you're in. Yeah, so in this case, if you wanted to control for them, you would see a main effect of environment, but not a main effect of genes, at least in this one environment. All right. So questions on this. We're sort of hammering home this idea of gene-environment interaction and what that means. Take a look before we move on to epigenetics. All right, so we're going to move a little bit quickly because we're a little bit behind time. But so take a look down, number 3 down here, we've got epigenetics, right? And so our definition of epigenetics is just changes in gene expression due to structural modifications of chromatin. So epigenetics is a change in gene expression due to structural modifications of chromatin. And it's not epigenetics if it involves a change in gene sequence, right? So we're not talking about changes to the actual nucleotides, and we're also not talking about transient things like transcription factors, promoting transcription of a gene. But we're talking about structural modifications of chromatin, and chromatin is just the genes and all the proteins sort of associated with the DNA. All right, and then we have two mechanisms that, at least on B side, these are the two mechanisms that we care about. They're acetylation and methylation, right? You might remember these from lecture last week, acetylation and methylation. Mnemonic I like is that acetylation amplifies expression, methylation mutes expression. So if you're adding acetyl groups, that's usually like loosening up the histones, making DNA more accessible, and you can express more with acetylation. And with methylation, we're usually compacting the chromatin. DNA is less accessible, and you get less expression. And just a caveat with that, like those are general rules. In reality, like it's probably more complex, and you have to look at complex patterns of acetylation and methylation. But for our purposes, that's a general heuristic that you can use to know what these different epigenetic mechanisms do. All right, so we talked about the sort of Pima Native American genotype as the thrifty genotype model. Now we're going to turn to the thrifty phenotype model, right? So this is down at the bottom. We have two different thrifty phenotype models. Both of these are about the nutritional environment of the fetus and the way that impacts the fetus's later metabolism in life. So anybody remember the difference? We've got the Barker thrifty phenotype hypothesis and the Gluckman thrifty phenotype hypothesis. Was there a difference between those that anybody remembers? Yeah, Corey.
So like Barker's hypothesis is that the fetus isn't getting a lot of nutrients, so it decides it's going to store
nutrition,
fat, but that's just for that moment in time.
Yeah, it's this immediate adaptation.
But Gluckman's hypothesis is the fetus is like, well, the reason I'm probably not getting like any nutrition is probably because like my mom lives in a like low nutrient environment, so for the rest of my life, like this is how I'm going to be. But
totally,
it's for like a sort of like a life course, and this Barker's more for like that immediate moment.
That's exactly it. Great job. And so Gluckman's, they basically have the same effect, that you have epigenetic mechanisms that give this developing person a thrifty phenotype. That's the same effect in both of them, but what's different is sort of the motivation, whether it's the immediate environment that like, oh, I need to survive with Barker, or if it's sort of this prediction of an adult environment with Gluckman. And so Gluckman is the one that we really look at and think about more, because that's the one where it matters what adult environment you end up in, right? So in this case, we're going to be looking at an environment-environment interaction, right? So again, we have three variables. We have our dependent variable of obesity, but then our two independent variables. One is the adult environment, right? Is it a low-fat or a high-fat adult environment? And then what's our other independent variable? So it's not genotype this time.
Prenatal nutrition.
Yeah, the prenatal nutritional environment. Oh yeah, it says it right there. So we've got poor prenatal nutrition and normal prenatal nutrition, right? So we'll say poor is in red and normal prenatal nutrition in green. And so, again, practice. Try drawing this graph out for yourself. See what you come up with for the low-fat environment and the high-fat environment for these two different genotypes. And again, we're going to be looking for an interaction, right? So we know that these lines aren't going to be parallel. All right, so if you have sort of normal prenatal nutrition, right? So this is sort of typically developing fetus who has enough nutrients in its environment. in the low-fat environment, low obesity, that's fine. What about in the high-fat environment? Compared to the poor prenatal nutrition, is it going to be a big increase or a smaller increase? Smaller, right? Great. Sounds like you guys got this. And then for the poor prenatal nutrition, if it ends up in a low-fat adult environment, that's great. That's what it was predicting, right? With Gluckman's model is about predicting the future, and it has no maladaptive consequences for obesity. But if you end up in a high-fat adult environment, there's this mismatch. You get like very high rates of obesity and diabetes. And so in this case, we have our environment-environment interaction, because those are the two independent variables that are interacting.
Will it usually look like the gene-environment interaction? So like the graphs, they look really similar.
Oh, yeah. Well, not necessarily. Like sometimes you might have, some other interaction that might be, like a crisscross. And so in this case, this is a really strong interaction where in one environment, you have completely different, like opposite outcomes than you would in another environment. So it can look different. You'll just want to be able to unpack that. So take a look at your notes and tell me like what questions you have about this material before we move on. Yeah, Lila.
Could you explain again what Barker's thrifty phenotype is about?
Yeah. So Barker's is about an immediate adaptation. So it's like the motivation for these thrifty phenotype changes is to sort of survive living in this nutrient-poor immediate environment, sort of like the mother's nutritional environment.
What do you mean by motivation? Because surely Gluckman doesn't think they're like consciously doing it.
Yeah, no, and in either case, it's not the fetus like thinking about it. It's just sort of like, why are these epigenetic changes happening? And it does seem a little silly to me too to call it like this motivation for like why it's set in a thrifty phenotype. But Barker came up with the idea that like, oh, it's just responding to the immediate nutritional environment and trying to survive. And Gluckman is like, it's actually foreshadowing this sort of adult environment and trying to set its epigenetic mechanisms to survive for that. Yeah, Corey.
So when it comes to like Barker and Gluckman, I know that like Barker's like his idea is like it's for the immediate moment, but like don't these effects like stay with the individual for like the rest of its life anyways?
Yeah.
So wouldn't that graph apply to Barker as well? This graph.
Yeah, it's just that like Barker wouldn't be talking so much about like a mismatch of environment, of future adult environment, but they do both apply. Like they have the same ultimate effect down the line. Yeah. Yeah, Lisa.
So when you're talking about this mismatch, it's because like they adapt to like store more fat when they have a poor prenatal nutrition, but then if they end up in a high fat.
Yeah, and that's the mismatch. Yeah, the mismatch between the prenatal sort of programming and the eventual adult environment. Yeah, Garcia.
So following Barker's hypothesis, if the person like changed environments, would they be able to change phenotypes to adapt to that environment, or only during developmental period?
Yeah, that's sort of the thing with epigenetics is that it's really about sort of very early life, prenatal and postnatal, like very, very young age is when these epigenetic changes are getting set for life. And so there's not much ability to change your thrifty phenotype. By the time you get older, you just have to change your environment, right? So if you end up in a high-fat environment, you don't eat all of the high-fat things available to you if you have your thrifty phenotype. All right. Oh, yeah, Corey.
And then for these epigenetic changes, can these like changes be passed down to like your offspring?
So that was sort of like this kind of controversy thing in the article that we assigned was like, is it clear that, they can be passed down to offspring or not? And it seemed like, it looks like they might in, this one rat model, but it's not like a lot of good evidence for passing epigenetic changes down. Because one of the other things that it mentioned is that early in life, pretty much most all of the epigenetic stuff gets sort of wiped, and it's sort of reprogrammed and set anew. So yeah. All right, so now with our last few minutes, take a look at this article. So you've got a scientific abstract here. And then with the person next to you, try to read through this abstract. You can kind of skip over the data sources, the study selection, and the data extraction. But with a partner, try to take a couple minutes, try to answer these questions about this study. Try to figure out what's the big deal with this study, what do these questions tell us, and then we'll come together. And maybe just for time, we'll just come together as a class and try to address this. So what kind of study is this? What do we call it when they take, they didn't do the research themselves, but they looked at all these other studies? What's that called? Meta-analysis, right? So it's not they're doing their own analysis, they're analyzing other people's analysis. So it's this meta-analysis. And we think of this as correlational or experimental? Correlational, right? Because we're looking at what's the dependent variable? What comes out of there? What are they looking at?
Mortality.
Yeah, so if we're looking at the dependent variable, it's mortality. Like how much are people dying? Yeah.
Can you have an experimental study and a meta-analysis?
Uh-huh. Yeah, you could do a meta-analysis of a bunch of other experimental studies. So it's like the meta-analysis itself, you don't assign other studies to like a condition or not.
Still, because you're determining what the cause and effect based on other people's.
Yeah, but you could, like, in this case, it's like analyzing other people's correlational studies, but you could do a meta-analysis analyzing experimental studies as well. Okay, sorry, outcome variable is obesity, and what's the independent variable, the predictor variable? BMI, right? And so we're talking about body mass index. What do you think of as in terms of like a limitation of using body mass index as an indicator? Yeah.
We talked about how it doesn't differentiate between people who might have a lot of muscle mass versus a lot of mass in terms of fat.
Yeah. Yeah, like really strong, muscly people can be categorized as overweight or obese, according to BMI, because it doesn't take into account sort of the composition of your body. And what's the big finding here? Why did people, this study made like a big splash, big impact, reported in the New York Times. Why? What's so surprising about this? Yeah, Lila.
Like just normal overweight and then almost obesity, or like the first level of obesity are protective against mortality.
Yeah, right? Because like it says, so it reports these hazard ratios, and hazard ratio is basically, it sets normal at 1, like so, the number of deaths or percentage of deaths in normal is 1, and then the hazard ratio is compared to normal, do you have more or less mortality, right? So what was the mortality for the overweight group? What was the hazard ratio? 0.94, right? That's down in those results. And that means there's a 6% less chance of dying in the overweight category than in the normal weight category, right? And so that's sort of like, whoa, that kind of turns our ideas of sort of health and goals and things like that on its head. Like suddenly overweight seems to be protective in some sense, right? But in terms of like critiquing this study, you might want to think about, it's like what kinds of people are going to be falling into these categories, and right? And these are people from all over the world. So maybe in some places in the world, being overweight means that you're sort of wealthy enough to afford regular food that you could actually put on extra weight. And you might also think about like what happens when people die oftentimes is they lose a lot of weight sort of on their downward trajectory towards death. And so maybe they used to be in the obese or overweight category but lost a fair bit of weight as they died, right? And so maybe that overrepresents the number of dying people in the normal category or something like that. And then this last page you can take a look at on your own, but it's basically analyzing a little bit, this is the other separate sheet, analyzing a little bit of the healthcare costs and things like that associated with smokers, obese individuals, healthy living individuals. And then depending on how you break it down, it could be more or less for a given category. All right, and then the last thing is on the back. This is sort of a practice short answer question that you can take a look at on your own on the hypothalamic feeding circuits. And in terms of the outcomes for today as we move towards the midterm, what you want to be able to do is, one, compare Bronfenbrenner and Hertzman models to sort of a developmental question, like what effects do these different things have. You want to be able to analyze these graphs to determine what main effects are going on, what interactions might be happening. You want to understand how epigenetics is an example of gene-environment interaction and be able to distinguish between Barker and Gluckman's thrifty phenotype hypotheses. You want to be able to like take a look at an abstract and pull out, meaningful information, understand what it means. And then after you do this last page, you'll want to be able to sort of understand how the hypothalamic feeding circuit works and all these different feedbacks. All right? Okay, thanks a lot, guys. I'll see you next week.